All right, part two, we're going to now turn to when multiple genes can affect the same character. So an example of this is actually in snakes, so corn snakes, skin coloration in corn snakes. And there's 4 different colors that the snake can have. So the snake can have camouflaged, black, orange, or albino. So this should be a first clue that we're seeing something different than before, because we see 4 different traits for a single character. So again, there's a single character, which is color, skin color, and there are 4 different variants of it. So we saw before how you could have multiple alleles. So potentially what we're seeing is something similar to the Himalayan rabbits, where they have 4 different colors caused by 4 different alleles. But in this case, what we're actually going to find is that it's caused by 2 different genes. And so what are some ways that you can know that it's actually caused by 2 different genes? So the genotypes that result in these different colors are shown next to the pictures of the snakes. So we have 2 genes, an O and a B, and we have 2 alleles. There's an O plus and an O, and a B plus and a B. And so what this means, this is an indication of dominance, so that O plus is dominant to O and B plus is dominant to B. But the way you should read this is that it means if one allele is O plus, it doesn't matter what the second allele is. It could either be O plus or O. If one allele is B plus, then it doesn't matter what the second allele is. So any genotype that falls within this is camouflaged. So there's 4 different genotypes that actually result in camouflage snakes. There's 2 genotypes that result in black snakes. So the O gene must be recessive O, so you must be homozygous for the recessive O. But then as long as you have a B plus, it doesn't matter what the other one is. So what are the 2 genotypes? So it could be OO, B plus, or OO, B plus, B. And then similarly orange, if you have little b, and 1 O plus, it doesn't matter what the second one is. And then albino can only be homozygous recessive of both. So let's say we did a cross between camouflaged and albino snakes, and they bred true. So again, this can only produce 1 type of gamete, O plus, B plus, and OB. And so what happens if we do an intercross between these? So first, the F1 will have to be heterozygote. It'll be O plus O, B plus B. So all of the F1 will be the camouflaged snakes. And now, if we intercross these further, there will be 9 camouflage, 3 black, 3 orange, and 1 albino. And so how is that possible? So you have to determine what are the gametes that can be produced by the F1. And what you'll find is that there are 4 gametes. And you'll end up having a 4x4 square. And then the exact same thing here. And so just like in the example of a dihybrid cross, I will let you do the actual Punnett square. The result is that you have a 9:3 to 3:1 ratio. And so if you group all of the camouflaged ones, what you'll find is that you have 3 different genotypes that can be camouflaged, or sorry, maybe 4 different genotypes that can be camouflaged, multiple ones that can be black, multiple ones that can be orange, and 1 that can simply be albino. And so this is different from the rabbits, because the rabbits, we never saw that 9:3 to 3:1 ratio. We never saw that ratio that is a function of 16ths. And so this is one of the ways you can tell the difference between is something caused by 2 genes or is something caused by multiple alleles of the same gene. And the way you you do the genetic analysis is one important clue is what is the ratio in an F2 intercross. And so the ratio in an F2 intercross, if it's a function of 16, usually that means that you have individuals that could produce 4 gametes each. And the only way that's possible is if there's 2 genes that have heterozygote alleles within them. So 2 genes affect a trait. Dominance recessiveness still applies. And now epistasis occurs when you also have the effect of masking of one of the classes. So this is described as the gene masks the phenotypic effects of another gene. But the way I think about it, and you can think about this in terms of something like Labradors, is where you lose an expected phenotype. So what do I mean by that? Okay, so we have Labrador retrievers, and coat color in Labrador retrievers is caused by 2 genes. There's a B gene and there's an E gene. The B gene has 2 alleles, big B and little b, and the E gene has 2 alleles, a little e, big E, and a little e. So let us say you have an intercross. So again, you have the heterozygotes cross to each other. So you do the Punnett square, and the first thing that you should always think about when you do something like this is that there are the following genotypes. And this is also true with, for example, the dihybrid cross and the snakes that we just saw. So this is just a ratio to start to memorize, the 9:3 to 3:1. And so because this is big A and then the second allele doesn't matter, big B and the second allele doesn't matter. So whenever dominance recessiveness occurs, then you expect all of these genotypes to share the same phenotype. So we saw this with the P's. So for example, if you had a dihybrid cross, you saw a 9:3 to 3:1 ratio, where the dominant trait, there was 9/16 of the offspring would have both dominant traits. And then 3/16 would have 1 recessive, 1 dominant, 3 would have the opposite. So this is just a good starting point to always remember, 9:3 to 3:1, and you should also be able to remember the genotypes that fall within these categories. So Labrador retrievers, if we do this intercross, one of the things that we find is that there's only 3 phenotypes, 3 traits that we see. So instead of there being 4 expected traits, there's only 3. So there's a black lab, there's a chocolate lab, and there's a yellow lab. And then the ratio is 9:3 to 4 instead of this 9:3 to 3:1. So what happened? Well, what happened is, for this case, for Labradors, these 2 classes are grouped together. And so what you'll see, as long as you have the recessive homozygous for the second gene, now it doesn't matter at all what that first gene is, okay? And so these classes end up getting grouped together. And so this is black, this is brown, and this both together is yellow. And so this is an example of epistasis. So epistasis not only means that you have 2 genes that affect a trait, but it also means that you have this masking effect. So essentially you lose one of the expected traits. So instead of having 4 traits, we only have 3 traits. And now there are also, you see things that are grouped together. So these classes are grouped together, and so the ratio goes to 9:3 to 4. And so there's actually a large number of ways that you can do this. So another example of this is in squash. So there's certain squash that have different colors. They can either be white, green, or yellow. And these, again, are the expected categories. So we have this first category that accounts for 9, this category that's 3, this category that's 3, and this category that's 1. And so in this example, we also see a masking effect. And what this means is that if you have one of the dominant W alleles, it does not matter what your G allele is. You are always white. And so in this case, the resulting ratio that you see in an intercross is a 12 to 3 to 1 because these two categories are grouped together. So epistasis can manifest in a number of different ways. That can either result in 9 to 3 to 4, it can result in 9 to 4 to 1. There's all sorts of ways you can combine these categories together. So whenever you see that, that's epistasis. So we end up naming some of these epistasis. So there's actually names for lots of them. The only two that I'll expect you to know, one is recessive epistasis. So the Labrador retrievers are an example of recessive epistasis, and then dominant epistasis. So dominant epistasis is an example from the squash. And so recessive epistasis occurs when the recessive allele at one locus masks the effect of the second locus. So why are Labradors an effect of that? Because if you are recessive genotype of e, it no longer matters what your big B is. You are always a yellow lab. So it masks the effects of the other gene. And then dominant epistasis is if you're dominant for a certain gene, so if you have the dominant allele, then it masks the effects of the other gene. So it no longer matters what this your G genotype is, you always have the white color. And so dominant epistasis is that situation. So recessive epistasis has this characteristic 9 to 3 to 4 ratio, and then dominant epistasis has a 12 to 3 to 1 ratio. So these are just the ways that you use genetic analysis in order to understand certain fundamental genetic relationships for a trait. And there's really a large number of different types of epistasis that are possible. So we have covered recessive epistasis, we have covered dominant epistasis, but there are other ways to group things together as well. So an example in peas with flower colors is these three categories. Anything that has a recessive has the same phenotype. So we group these all together as having the same color. So in this case, when you add the 3 plus 3 plus 1 together, the ratio that you get is 9 to 7. Squash fruit shape can either be disc, sphere, or long. And the genetics for this is that you group together these two categories. And so you get a 9 to 6 to 1 ratio. This is an example of how to get a 13 to 3 ratio in chicken. So these are all real life examples. So you can see that genetics is often very complicated. All of these cases are out of 16. So that's one thing to remember, is that all of these cases are out of 16. So if you add 9 plus 3 plus 4, 12 plus 3 plus 1, 9 plus 7, etc., all of these add up to 16. And so that's one of the ways that you can recognize pretty rapidly if something is probably caused by epistasis, or if something is caused by two genes, is because the ratios add up to 16.